Friends, Namaste. Welcome to Dr. Varsha Gaitonde's channel. And in this topic, I'm going to talk about trans, cis, and intragenesis. That is transgenesis, cis-genesis, and intragenesis. Let us start. First, we'll talk about transgenesis. As you know, in transgenics or genetically modified organisms, means the gene is taken from some other group. As you can find out that the gene pool is divided into four groups like gene pool 1, gene pool 2, gene pool 3 and 4. To refresh your knowledge, gene pool 1 means the varietal or the species compatibility, how oftenly they can be crossed. Means in gene pool 1, the crossability is very easy. When we move about gene pool 2, the crossability is quite different. That means 50-50% crossability can be obtained. That means 50% chances of crossing and 50% not getting crossed. Means we may observe some of the barriers. For example, if we cross the wheat and its wild ancestors, that means some of the wild ancestors may be compatible with wheat and some may not. Gene pool 3 means they are not at all compatible. That means whenever we talk about the related genera, they are not compatible. For example, if we cross sorghum and bajra, that is compatible, we can call it in it gene pool 2, or we may also place some of the varieties in gene pool 3. But when we talk about wheat into rice, they are not at all compatible, even though they are belonging to Poaceae family. That is nothing but gene pool 3. All the transgenic plants are classified in gene pool 4. Gene pool 4 means they are taken from some other groups. For example, I may say that one gene from chicken, for albumin of chicken, that was transferred to human. That means for protein content the gene is transferred. Likewise, when for different particular trait or different traits, like it may be for nutrition, the popular example is papaya ring spot virus. For ring spot virus, the gene was taken from some other species and introduced into papaya. Then other popular examples like glyphosate resistance, then bt cotton, and salt tolerance in rice and all those things. That means the transgenic approach is popularly known for us because of bt cotton. Okay, as we can say, in India the commercially cultivated only bt or the genetically modified crop is bt cotton, and that is a hybrid one. So what is the mechanism followed in bt or transgenics? Here the gene is taken from some other species, that is, or some other genera or anything, and that is introduced or remodified by constructing a vector. That vector will be introduced into a plant, and with the help of agrobacterium or any other technique. Agrobacterium is a biological or natural technique where the tDNA is multiplied into n number of times by the quick process or quick multiplication of agrobacterium. Then that multiplication inside the plant, and here the nature of agrobacterium tumefaciens is tumor inducing, but that tumor inducing portion of that particular vector construction is silenced, and the rest of the portion of multiplication is enhanced, and likewise that particular gene will be distributed throughout the plant body. That is all about transgenics. Transgenics का मोटा मोटा मतलब होता है gene may be taken from any group, may be from some other genera, family or anything. Okay, let us move to cis-genesis. Cis-genesis and transgenics are intragenesis are quite different from transgenics, because cis-genesis means here the extra DNA is taken from the same family or the same group. In cis-genesis, as it in transgenesis, extra DNA is stably built into the plant DNA. Major difference between transgenics and cis-genics is the origin of DNA. Okay, origin of DNA means here we are taking DNA from the same group or same genera. Here the extra DNA originates from a plant with which the acceptor plant, that means the plant that will receive the extra DNA, can crossbreed. That means this can be placed, the plants which are crossable can be placed under gene pool 1 or 2. Cis refers to within the same crossable group. Example, adding tomato genes into tomato. Cisgenesis aims to obtain the same result as through conventional crosses, but in a much faster and more efficient manner. You may be wondering that even though the mass technology or marker assisted selection, mutagenesis, and many approaches are popular in India, but still why should go for cisgenesis? Cisgenesis will give the fast application. That means when we go for breeding, we should use more number of markers than more effort for many number of generations. But if we go for cisgenesis, means already you can feel that in many of our laboratories the transgenic approach is popular, because even though the varieties developed were not in the market, but the approach is quite familiar with some of the scientists, so they may go for cisgenesis and utilize this technique for the betterment of society. So when they are taking the same gene from same plant and introducing, that means less time and efforts. Wild species of potato from Mexico and Andes have genes that make them resistant to disease, that is for blight and all those diseases. Then the genes can be transferred by crossbreeding to potato cultivars. But this process is complicated and takes time. With crossbreeding, undesirable traits are also inherited. In order to comply with the definition of cisgenesis, the genes that are added must also come with their original regulatory unit, the switches that decide when a gene is activated or silenced and to what extent. That means here the promoter or the cassette, we may say the regulatory cassette is designed here, like how the promoter should be, how the activator, how the regulators, all those things should be, not by modifying them. As such they are transferred. That means a gene is transferred as such it is in the other plant. That is nothing but cisgenesis. All the hereditary information for a particular trait is therefore fully incorporated, and the gene has the identical sequence including introns to the crossable plant it comes from. Cisgenic plants may not include bacterial selection gene such as antibiotic resistant genes or selection genes from non-crossable species. Selection genes can be used during the process of genetic modification, but they must then be eliminated from the plant DNA because natural crossbreeding barriers are not crossed. Cisgenesis has wider acceptance among the public. From the point of view of environment and food safety, there is no difference between cisgenesis and transgenesis. It is always the trait for which the introduced gene encodes that determines whether a crop is safe or not, whether that gene comes from the same species or a crossable species. Distinction between cisgenesis and transgenics may be important depending on the legislation that varies across the world. Certain applications of cisgenesis may or may not be regulated. Coming to intragenesis. Intragenesis is just opposite to transgenesis, or we may say it is a modification of transgenesis. In intragenesis, certain DNA fragment is stably built into the plant DNA, and again DNA is used from a crossable species or same species. ये बहन जैसी है सिसजेनेसिस की. सिसजेनेसिस में क्या होता है, कोई भी जीन उसके रिलेटेड स्पीशीज या रिलेटेड प्लांट से लेंगे एंड दैट इज जस्ट इंट्रोड्यूस्ड और गिवन टू ए कल्टिवेबल प्लांट. दैट इज सिसजेनेसिस विदाउट एनी मॉडिफिकेशन. ठीक है. लेकिन इंट्राजेनेसिस में क्या होता है, हियर द जीन इज मॉडिफाइड. दैट मीन्स द रेगुलेटरी कैसेट इज चेंज्ड अकॉर्डिंग टू आवर इंटरेस्ट. Just like cisgenesis, the specific type of genetic modification is done. Difference is that the original composition of DNA fragment is not retained. ये आपको याद रखना है. The original composition of DNA fragment is not retained. With intragenesis, new combination of existing DNA are made. The most common case is that gene X is combined with the gene switch of gene Y, means the promoter may be from some other gene and regulatory may be from some other gene. This kind of modification is nothing new since many organisms have undergone spontaneous DNA rearrangements during evolution, resulting in the formation of new DNA combinations. In nature, this occurs at random and the individual that undergo undesirable rearrangements disappear. With intragenesis, the elements of chance are removed. Intragenesis is an interesting breeding method to allow a certain trait that already occur naturally in the plant, example, protection of certain vitamins in the leaves, to be expressed in the harvestable part, example, seeds as well. So here I have a diagram to give you a complete picture of transgenesis, cis-genesis and intragenesis. As you know, in case of transgenesis, some gene is introduced, okay, from some other group. Here the gene is introduced. When the gene is introduced, then for introducing a vector is made. Vector is having left border, right border, then many of the modifications will be there. There will be some regions for resistance and all those things. The tumor causing part will be disabled and all those things. After constructing this vector, where the gene should be, where the other promoter should be, it is all casseted or it is all fixed and made into a one construct, and then it is introduced into a target host plant where it need to be multiplied. After that, by using selectable or scoreable markers, the transgene or the part or the promoter or any kind of that particular gene which is having this particular tDNA of foreign DNA or foreign gene, that is selected and further it is modified or it is multiplied by keeping it in the agrobacterium. That is all about transgenesis. When we talk about cisgenesis, as such, here we have a gene of wild, okay, and that is transferred to a cultivable plant. Here promoter, coding sequence and terminator all are from the same gene of that particular wild variety. But when we talk about intragenesis, here you can see the promoter is from gene 2, coding sequence is from gene 1 and terminator is from gene 3. That means there is a difference in the parts of that particular gene or the regulatory material what we take for the expression of that particular gene. The parts are taken from different genic combinations. To summarize this, all the three techniques, it may be transgenesis, cisgenesis or intragenesis, should be regulated, because whenever we are taking genes from other plants or other thing, means they may be causing difference in that particular organism. So these are regulated, but the regulatory mechanism is different, because transgenics has gene from some other plant, but in cisgenesis and intragenesis that much regulations are not required. So these two techniques, cisgenesis and intragenesis, can be utilized for the betterment of our plants and to hasten and fasten the resistant mechanism, as well as for the development in other aspects like biotic or abiotic stress. I hope this topic is helpful for you. If you like this video, please do not forget to subscribe and like, keep on sharing and commenting. Thanks once again.